Genetic manipulation. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. Within aquaculture, genetic manipulation is emerging as a powerful method for breeding of fish and shellfish and for developing alternative sources of feed and vaccines to combat diseases. Genetic manipulation, commonly known as genetic engineering or genetic modification, is the direct manipulation of an organism's genes using biotechnology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved on novel organisms. Genetic engineering is accomplished in three basic steps. First, is the isolation of DNA fragments from a donor organisms. Second, is the insertion of an isolated donor DNA fragment into a vector genome. And third, is the growth of a recombinant vector in an appropriate host. One of the known genetically modified fish is Atlantic salmon. The idea of altering Atlantic salmon DNA to get the fish to grow more quickly. So how do you get a GM fish? The DNA of a Chinook salmon is combined with DNA from the ocean pout. The Chinook salmon DNA make the salmon bigger and the ocean pout DNA is used to add resistance to cold temperatures as the ocean pout has many freeze proteins on its body. A salmon's growth hormone are more active during certain times of the year. For the growth hormone to stay active all the time, a DNA taken from a fish called an ocean pout which produces a special protein all year long that helps it survive on frigid waters. The DNA of ocean pout keeps those proteins turned on and running and connected it to a semen growth hormone gene which have the effects of keeping growth hormone on. As a result, the genetically engineered salmon grows twice as fast as regular salmon when actually consuming less feed. The difference is significant. These fish are both about 2 years old. They have the same age and there's such a huge size gap between the two of them. The product is safe to eat, has no significant impact on the environment, and it found no biologically relevant differences between salmon and other farm-raised salmon. Another is the glawfish. The gene is taken out from sea coral and as soon as the egg fertilized, the gene is added to the egg and it enters into the DNA. This fish were originally developed as a kind of early warning system of toxins in water. The idea is that you drop it in a bucket of water taken from a river, and if the water is polluted then the gene would switch on and the fish would start to glow. There are couple benefits and risk when making genetically altered fishes. The benefits include consumption of genetically modified specimen reduces stress on natural waters. Fish reportedly taste as good or better than wild fish. More sustainable than catching and or farming wild salmon. Risks. The risks include lacking of information on the long-term effects of eating genetically modified organisms. If the genetically altered fish were to escape into the wild, they could mate with wild fish causing genetic contamination. There is also a risk of pollutants/contaminants in the facility. That's all, and thank you for watching.